Welcome to Biology Made Easy. Today we are discussing the processes of crossing over and independent assortment of chromosomes. This process takes place in meiosis, and so in effect we'll be discussing meiosis. But the reason why we are discussing this is not only meiosis, but the fact that no two individuals of any species are the same. The major cause is crossing over and independent assortment of chromosomes. Now, meiosis takes place in specific cells in all organisms. In human beings, meiosis takes place in the males in the testes, in special cells called spermatogonium. In the female, it takes place in the ovary, in ogonium. In plants, it takes place in the anther to produce the male sex cells, the male gametes. In the female, in the ovary, to produce the ovules with the gametes. So, meiosis in all individuals has specific cells it goes through, and meiosis produces gametes. You have one body cell and it produces 4 different types of cells with different combinations of genes. And when crossing over comes in, then the number of different combinations of genes becomes big. And this process we are going to show today shows it. Now, we are saying that crossing over independent assortment is the major source of genetic variation, but other reasons are, fertilization is a random process. You have one egg surrounded with thousands of sperms, and each of the sperms is different, and this one sperm is going to fertilize an egg. Fertilization is a very random process. Getting a mate too is random. Who knows Ken will marry Ajua? Very random. Mutation also brings about some variation, and then the environmental effects on the organism itself. The environmental effects on genes, we discussed it in variation, and then there's also environmental effects like nutrients, etc. So, no two individuals of any species are the same. Well, process of crossing over and independent assortment in meiosis. Meiosis is a type of cell division. In meiosis, one cell produces 4 cells, and each of the 4 cells have half the number of chromosomes that is in the original. So we start with one cell, it has 4 chromosomes. Meiosis will produce cells, and it will have 2 chromosomes, half the number of chromosomes, and they are the gametes. So what happens during the production of the gametes is what we are going to look at that brings about genetic variation. We are starting with a cell. This is a body cell. This cell is going to go through meiosis, and we have 1, 2, 3, 4 chromosomes in this body cell. Now, these chromosomes, when the cell is not dividing, is chromatin material. Then, when the cell starts preparing to divide, the DNA replicates. This is the homologous chromosome, these two. One is maternal, one is paternal. These two are also homologous chromosomes. Now, these are the chromosomes I've brought here. We have this chromosome. I have labeled some genes on. Gene big A, big B. The other homologous chromosome has gene small a, small b. Then I have this chromosome, gene big D, big E. And then this has genes small d, small e. These cells, as it begins division, there are spindle formed. So these two are homologous chromosomes. These two are also homologous chromosomes with their genes. Now, you can see the chromosomes well because the chromosomes have undergone DNA replication, but you can't see the chromatids well. As prophase goes on, you can see the chromosomes well. You see this chromosome? with big A, big B here, you see this chromosome as two sister chromatids. It's still the same gene big A, big B, gene big A, big B. So it's simply two sister chromatids. This is one chromatid, this one chromatid, held by a centromere, gene big A, big B, big A, big B, because it's the same one chromosome. This small a, small b, held by a centromere. But the chromosomes usually are not seen as straight lines like this. They are like this. That is why we are here. So we have this chromatid, big A, big B. We have this chromatid, big A, big B. It's homologous chromosome. You have small a, small b, small a, small b. Then we have this chromosome. It's seen well as sister chromatids, gene big D, big E. So as sister chromatids, you have gene big D, big E, big D, big E. The homologous chromosome, you have gene small d, small d, small e. So this is one chromatid, another chromatid. So this is how the chromosomes are, early prophase. Homologous chromosomes lie together, and the process of them lying together like this and like this is called synapses. Process of them lying together. And the homologous chromosome lying together, they form bivalents. So this one bivalent, this one bivalent. Now, after this process, crossing over is going to take place between non-sister chromatids of homologous chromosome. So this is one homologous chromosome. This and this are sister chromatids. This and this are sister chromatids. This and this are non-sister chromatids. And crossing over is going to take place between these two so that the genes will be exchanged. Crossing over is going to take place between these two non-sister chromatids of the same homologous chromosome. That is what happens. Crossing over can take place between any genes and any parts of the chromosomes. And when genes are farther apart, crossing over between them becomes easier. So by the middle of prophase 1 in meiosis, crossing over is taking place between non-sister chromatids of homologous chromosomes. So you see, where the crossing over is taking place, the chromatids try to come together but still repelling. But yet there's a point where the chromatids kind of join, and then there's a break there, and the exchange takes place there. The place where there are kinds of break points are called chiasmata. One is chiasma. So you see here, there's a chiasma taking place here. Here too, the non-sister chromatids are coming together, but they are also trying to repel this one and this one. And that is the point that it will break to exchange parts. That crossing over is taking place there between non-sister chromatids of homologous chromosomes. Crossing over does not take place between different homologous chromosomes. So by the late prophase 1, crossing over has taken place between non-sister chromatids of homologous chromosomes. So you see, we started with this chromosome having this chromatid, gene big A, gene big B, and this one gene big A, but it now has gene small b because of the crossing over. Then this one too, we have gene small a instead of having gene small b here. Because of the crossing over, we have gene big B here. And this one is still gene small a, small b. The same crossing over took place between non-sister chromatids here. So we have this change occurring. Late prophase 1, crossing over has taken place. We now go to metaphase 1. And metaphase 1 is where independent assortment of chromosomes begins. Now, this chromosome and that chromosome are going to lie down at the equator as bivalents. And these chromosomes are going to line up. The way they are going to line up, they are irrespective of how these chromosomes are going to line up. And that is how the independent assortment starts. Now, you see this chromosome here, it can lie down in the same plane with this chromosome, or it can lie down in the same plane with the other chromosome. So we start like this. This chromosome is lying at the same plane with that chromosome, and so the homologous chromosomes will be here. The bivalents will be here. So this one is here, its bivalent is here. This is here, its bivalent is here. This is one arrangement that could happen at metaphase 1. We could have that arrangement, or this chromosome is going to lie in the same plane with that chromosome, and their bivalent comes down like this. Or that chromosome is going to lie in the same plane with this chromosome like this. Their bivalents are down. Or that chromosome is going to lie in the same plane with that chromosome like this, and their bivalents down. Now, notice the angle of the spindle in meiosis 1. This is the direction of the spindle. So the independent assortment at metaphase 1, you can have this arrangement, or that, or that, or that. Notice that this arrangement is the opposite of that, and this arrangement is the opposite of that. But then we have 4 different arrangements of bivalents with their other homologous chromosomes. So we are using only one of these arrangements to go through meiosis 2. So everything that is going to go through here is happening in all these other 3 cells. Phase 1, and we go to anaphase 1. And at anaphase 1, you have the homologous chromosomes separate. So this chromosome is going to go up here. That chromosome will come down here at anaphase 1. This chromosome will go here. Its homologous chromosome will go down here. This is anaphase 1. Homologous chromosomes separate. Then, after anaphase 1, you have telophase 1, where the cell divides into 2. And so you have 2 cells in telophase 1. So if I draw a line across it like this, I have telophase 1. Then we go to prophase 2. Prophase 2 is here. We see these chromosomes, they are in this cell, and these chromosomes are in this cell. Prophase 2. And in prophase 2, look at the spindle in prophase 1 and meiosis 1. Look, this is the direction of the spindle. Now in meiosis 2, starting from prophase 2, the spindle is at right angles of the original spindle. So the spindle is at right angles. So we have prophase 2 where the chromosomes are in the cytoplasm, not any kind of arrangement. That's prophase 2. Then you have metaphase 2 where the chromosomes are lying at the equator in a single line. So you have this chromosome and that chromosome forming a single line, and that single line in the equator. Then we have anaphase 2. At anaphase 2, centromere holding the sister chromatids break, and sister chromatids separate. So this chromatid and this chromatid are going to separate. So one chromatid is at this pole, the other chromatid goes to the other pole. That's anaphase 2. The same thing happens in this chromosome. One centromere breaks, one chromatid goes to that pole, another chromatid goes to this pole. The same thing happening in this cell. Centromere breaks and sister chromatids separate into different poles. Then you have telophase 2 where the cell divides into 4. And you see we have 4 cells, each with 2 chromosomes. These chromatids finally become chromosomes again. So we have 2 chromosomes in each. And if you look at the genes on the chromosomes, this is gene big A and big B chromosome, big A and big B. And this chromosome is here. And if you look at the arrangements of the chromosomes, you will see we have 4 different cells with 4 different arrangements of genes. Now, please note something, that from metaphase 2, we are going to anaphase 2. And the way the sister chromatids separate to go to different poles, You see we have this chromatid big A with genes big A, big B coming here, and big A, small b going to this pole. We had this one come into this pole and this one go to this pole. But we could have had this sister chromatid, this one, going to the same pole with this. Why not? Why this doesn't go with this? It's possible. All the possible arrangements is possible. So there's indication that some sorting out also goes on in anaphase 2, where the sister chromatids separate and go into other poles. So this is what happens, and we started with 4 chromosomes with these kind of genes on, and we said at metaphase 1 there's independent assortment. You can have this situation or that situation or that situation or that situation, and each one of these situations goes through anaphase 1, prophase 2 till telophase 2, each one of these. And so by the time you have 4 different types of cells, there's a possibility of having these chromosomes. These are chromosomes in the cells. So if we work out all the different arrangements that we could have, we can have 16 different types of gametes in this kind of crossing over. If we have one cell, that gives us 4 different cells. After meiosis, there's a possibility of having 4 different arrangements of these genes. And in total, we can have 16 different gametes in this kind of crossing over, where you have crossing over and independent assortment of genes. And no wonder, no 2 individuals are ever the same, because if you talk about human beings, we are not talking about 4 chromosomes, we are talking about 46 chromosomes, 23 homologous pairs, and a number of different crossing overs, plenty, because the genes are plenty. So we can have gametes that are very different. A couple, a man and a woman, have 20 or 40 or 50 or 100 children, or even 1,000 children, if it's that way possible. None of them will be the same as the other person or as the parents. Well, this brings us to the end of our discussion on crossing over and independent assortment. Please give us your likes, and then please do subscribe and add your comments, please. Thank you very much. Goodbye.